Good afternoon, everybody. This talk is about ash dieback as a conservation biology challenge, and is a team effort between Bayreuth Botanical Garden and the Swiss Federal Institute of Technology. So ash is a common and often abundant tree species. Here is an example from Switzerland, but also in many other European countries this is the case. For example, in Britain, ash is found in all 10 by 10 kilometer squares of the country. And so more is at stake than if a rare species were threatened. Normally, conservation biologists focus on rare species, and if they now start focusing on abundant species, it means that it is a real crisis. Also because not just ash is threatened, but also all the ash-associated species. And this is the case on a large scale throughout Europe now. So it's not just a conservation biology challenge, but a conservation biogeography challenge. That this is a huge problem for conservation biology was highlighted at the European level in this newsletter, Science for Environmental Policy. In the rest of this talk, I'll briefly introduce some aspects of ash dieback as a conservation biology challenge. So it is becoming clear that this is not just a problem for forest pathologists or for foresters. We need interdisciplinary collaboration with other disciplines because the consequences for biodiversity of this problem will also be affected by how people react to the disease. For example, for chestnut blight in Italy, a recent paper in Landscape Ecology showed that more chestnut trees were lost due to people felling them in reaction to the arrival of the disease than due to the disease itself. And a similar thing could happen for ash dieback, actually. So we know that interdisciplinarity is needed, but this is often easier said than done because people have different interests. And so, for example, if you ask a forester, he will tell you that we need to save money by cutting the ash trees before the value of the timber is reduced. But if you ask a conservation biologist, he will tell you that ash trees, even if they are infected, they need to be kept in the forest unless they pose a risk for safety, because many organisms are dependent on these trees. And I would also like to say here that last week I was in Brussels at a conference to identify ways to improve research careers throughout Europe. And 85% of the participants to this conference said that there were not enough opportunities to do research in an interdisciplinary way in Europe. Now, the problem with ash dieback is that also from a monodisciplinary point of view, there hasn't been much research. So we have a lack of knowledge for many groups of organisms associated with ash about their numbers, which species they are, and their biology. So that it is difficult to preserve them if we don't know about them. But some figures are started to appearing. So there are hundreds of lichens, of fungi, and of insects which are found on ash. Some of them are specific of ash, and so the loss of ash trees throughout Europe will pose a big problem to preserve these species. A key factor for preserving ash-associated species is how many of the ash trees will die. And so here we have two scenarios based on real data from Gotland Island in Sweden. You have on the y-axis the proportion of species that they are likely to go extinct. And the likely scenario is that all the infected ash trees will eventually die. And this is on the right with the field symbols in black. And so in this case, the proportion of species going extinct is higher than if just infected trees will die. So the other factor that is important is whether ash-associated species are likely to be able to survive also on other trees than ash. And so for many species, this is a knowledge gap that we have and that needs to be filled. But based on the data from this study, you can see that this proportion of species going extinct is higher if these species are only going to be able to survive on ash and not on other tree species as well. So for example, if you look at lichens associated with ash in Britain, here are some examples, just arriving at the letter C of the genus. And there are some acronyms. For those of you who don't know about them, NS is nationally scarce, EN is endangered, BAP is subject to a biodiversity action plan, IR is internationally relevant, then we have double D, which is data deficient, VU vulnerable, and so on and so on. So all these lichens are actually of conservation relevance. And there was a recent study published in Conservation Biology trying to assess how specific the assemblage of epiphytes is for different species, tree species found in Britain. And it turns out that species found on ash are similar to those found on oak, which is good news because oak is abundant, although not for all age classes. But epiphytes found on ash are also similar to species found on elm, which is bad news because we've lost many elms from the landscape due to Dutch elm disease. And also sycamore, Acer pseudoplatanus, has an epiphyte assemblage which is similar to the one found on ash. And so this is controversial because in Britain at least, sycamore is not well regarded by conservation biologists because it's supposed to be exotic and invasive. But it could actually play a rescue role for many species associated with ash. And so more research is needed using this approach for other kinds of ash-associated groups. Now, one point that has been made in the literature is that ash dieback could be temporarily a positive thing for biodiversity because it could increase the presence of forest glades, which is a species-rich but threatened ecosystem, because forests have become darker. And this might be true, but at the same time, there could be the loss of forest interior epiphytes, as has been shown already by a study in Sweden. And the other thing is that these forest glades are going to be temporary. Other tree species are likely to fill the gaps. And this could happen not just with native tree species, but also with exotic tree species, like Douglas fir, which has shown to have an invasive potential in Europe. And there are many other exotic tree species that could fill these gaps, and they are not very good from a biodiversity point of view because they are exotic, and so the species associated with them are likely to be fewer than the number of species associated with ash. One other similar point that has been made in the literature is that ash dieback could be positive for biodiversity conservation because it will increase the presence and the abundance of dead wood in forests. And this is also a threatened habitat, but very important for many organisms. And so there is a view that you have a pristine ecosystem, then exotic tree pathogens come in, and then you have a degraded ecosystem that you can try to restore. But in this case, you don't start from a pristine state because dead wood has already been reduced by foresters throughout much of Europe. And so what actually is supposed to happen here is that the exotic tree pathogen is helping to restore the ecological value of the ecosystem because it produces dead wood in huge quantities. One problem is here that we know that dead wood is needed from a variety of tree species, not just from ash, because organisms dependent on dead wood need a diversity of dead wood sources. And the other problem is that the dead wood production by ash dieback will just be temporary. And so since also young trees are affected, there will be a surge in the production of dead wood, but then a lack of recruitment. And so it is unlikely that dieback will continue to produce that wood in the coming decades. And so we need to take a long-term perspective on these kind of issues. And unfortunately, most of the studies modeling tree mortality in the literature are actually based on just a few years of data, not on this long-term perspective. So a few words on dieback also as a network epidemiology challenge. We've heard this morning that the trade in plants, and particularly of ash saplings, has contributed to spreading this disease across Europe. The problem is that we have little information about these plant trade networks, and the structure of these networks can make a huge difference to the likelihood of spreading these diseases. For example, we've heard this morning that there were many plants moved from northern Germany to Austria, and this is a long-distance link, which makes the network a small world. And it is easier for this kind of diseases to spread if you have this sort of long-distance connections. But we don't have information about whether we have a scale-free structure, which means if there are superconnected nodes. These superconnected nodes greatly facilitate the likelihood of spreading this kind of diseases. But unlike with human and animal trade networks, we still have to show that there are these superconnected individuals in plant trade networks. It would also facilitate the control, because if control were targeted towards these superconnected nodes, it might be more efficient and effective. So also this morning there was already some talk about the interaction between climate change and global trade, increasing global trade of plant material. This interaction is likely to make this kind of plant health emergencies more frequent in the future. But dieback shows that climate change is not needed, because we already have this problem just by the introduction of plant material. Since the disease spread towards the north, toward the south, in all directions, there's no role of climate change. And so just having global trade is enough to create these kind of problems. And maybe a few words also on assisted migration, which is a measure proposed in conservation biology to help plant species cope with the rapid climate warming which is predicted for the coming decades. There is a lively debate in the literature, but very few people have raised the issue that assisting the migration of plant species might actually result in also spreading their diseases, a bit like happened with dieback. So one problem is also that most of the literature on exotic tree pathogens is focusing on just a few of them, and we have very little knowledge about most of these exotic pathogens. At the moment, they are not causing trouble, but with changing environmental conditions, they might become issues similar to dieback. We don't know. And so, as a precautionary measure, more research would be needed on these pathogens, just to know, for example, the pathway with which they are associated. So, to conclude, dieback as a conservation biology challenge, the problem is that dieback is happening at the same time as many other threats to biodiversity, and so we need to maintain the host genetic diversity also for the host and for the associated species to be able to cope with these other challenges like forest fragmentation, climate change, and pollution. Particularly worrying is the loss of ancient ash and woodland. Vikki Bengtsson will talk about this in the next talk. But so one general recommendation is that dead, dying, and infected ash trees should not be cut unless they pose a risk to safety. And so dieback might temporarily result in a few benefits for biodiversity, but these are likely to be temporary, and so there are many deleterious effects as well. And so the most important benefit for biodiversity conservation would be to take this opportunity to improve plant biosecurity. I would like to thank my collaborators, in particular Anne Gross and Thomas Sieber from Zurich. Thank you very much for your attention.
Thank you very much, Marco. You've been very concise. We've 5 minutes. We will save these 5 minutes and go on to the next talk, Vikki Bengtsson.
Thank you. Good afternoon. The graveyard shift after lunch. I realise I don't sound very Swedish because I'm not. And just very briefly, I think I feel like a black sheep here today because I'm not a proper researcher. I work actually as an H conservationist, and what I'm going to present to you this afternoon is some work that we've done in Sweden looking at veteran trees. And I'd just like to mention that ancient trees, there's several of people in the room here from the Ancient Tree Forum, including David and Nev here, and ancient trees are something that we're very passionate about and very concerned about with regard to plant health on a large scale. Now, I'd just like to also mention Anna, who's my colleague here, who's the statistician. So don't ask me any questions about the statistics. I'll take them home with me. So, no. Okay. So just a little bit of background. I've worked in Sweden for the last 10 years and primarily worked with issues surrounding ancient and veteran trees. And in Sweden, there's an action plan for veteran trees, which was produced in 2004. And the primary objective of that was to establish the resource, the veteran tree resource in the country. So it involved surveying an awful lot of trees. We currently have a database with around about 350,000 deciduous trees. And the definition of the trees that were surveyed are trees that are hollow and over 40 centimetres in diameter, trees that are over 1 metre in diameter, and old trees. Now, in essence, the old definition is used much less often because we don't know the age of many of our trees because they are hollow. But also trees that contain threatened species were included in that survey work. It's a huge effort that was a big, big investment by the government in Sweden, and it's a fantastic resource of information about the current situation with regard to the old tree resource that we have. Now, the county in which I live, Västra Götaland, there's about 25,000 veteran trees being recorded, of which 17% are ash, and this is an example of one of them. So the background to the work I'm going to talk to you about today is that in Sweden, as in this country, we understand that our old ash trees have a very valuable role to play in biodiversity. They have lots of species associated with them, both specialists and generalists, as Marco has mentioned earlier. And they are culturally significant as well in Sweden, and I know that's the case here too. Many of them are pollarded or have been pollarded in the past. Now, in Sweden, there are also subsidies given for management of ash and other tree species in the kind of, if you like, the agricultural environment, so the wood pastures and the wooded meadows, where many of these old trees are. There's actually environmental subsidies for landowners to cut their trees on a regular basis. And we actually, in terms of ash dieback, have relatively little information about how it affects our very old trees. Now, I'm always delighted to hear that there's more work going on all the time collecting data about the older trees in the environment to help us understand what's happening. But I think we felt, sitting at lunch one day, we don't know what's happening with ash dieback and our old trees. Ought we not to be doing something about this, or at least trying to follow what's happening? And the other thing is that most of our veteran ash trees are actually outside of woodlands. The very old ones are old pollards, they are old wood pastures or wooded meadows. So this monitoring programme that I'm going to tell you about was set up in 2009 by a local authority. Now, I say that because that's pretty significant in terms of the level of investment. This was a small project where we scraped together budget from 3 different places to try and get out there in the field looking at some of these trees. We chose a very simple method, and there was no laboratory analysis based on field symptoms, and it wasn't intended as a research project. It was intended as a method to put in place to just keep an eye on what was happening with the old tree resource. And this is an example of a fantastic site in the southeast of Sweden called Mistefalls. It's a wonderful wooded meadow where the trees are still pollarded. And seeing this kind of thing was breaking my heart. And just to give you a very, I don't know if the pointer works here. Is that the pointer? No. Oh, well.
Switch it on.
switch it on, that helps. Yeah. Doesn't matter, I'll move over. This is...
You need the boy to do it.
Okay. But now what do I press? Yeah, I'm not sure actually. There, thank you. This is Sweden, the top's cut off, and this is the county of Västra Götaland, and this is Gothenburg, and Stockholm's up here. And ash grows basically here, roughly. And just to give you a sense, there's about 200 kilometres, what should I say, 130 miles, given that I'm in England, up top to bottom and east to west. And these two lakes are the largest lakes in Sweden, and this is the sea. So just to give you a sense of where. And the dots are the ash trees that we visited over the course of the period of this monitoring project. Now the method, well, I said to you we had about 25,000 veteran trees, of which 17% were ash. We did a random sample of those trees, of which included, now I'm interested in pollarding. I'm interested in old pollards, restoration of pollards, pollards in cycle. I am a nerd, happy to admit it. And I was very interested in understanding, because we were giving subsidies, whether pollarding, we should be continuing with it and funding it, or whether we should be stopping with it. So that was one of the things we were interested in. So we chose trees, according to our database, that were not pollards, maiden ash trees, and we chose trees that were pollarded within a recent history, within the last 10 years, and what we would call lapsed pollards that hadn't been cut for a much longer period of time. The same trees have been visited in 2009, 2011, 2013, in the summer, August primarily, chosen a very simple scale, which you can see for yourself, where 4 is dead and 0 is no sign of any kinds of symptoms. And you've seen lots of pictures illustrating what trees look like. And I think the epicormic flushing that you see in the crown is a very typical feature that you can see from a very long way away. So, some of the results, well, we were very clear to see that the whole county was affected. That was no surprise to us. And I think I'd like to consider myself an optimist, perhaps even slightly romantic about what old trees have to offer us in terms of the future of ash. Sadly enough, I'm not sure that my results support my optimism, but I'm still going to keep it until such time as I'm proven completely wrong. But what you can see is that we're increasing in the proportion of trees that are affected. So in 2013, we were up at 84% of the trees having some kind of visible symptoms. The thing that maintains my optimism is the mortality rate. Now, we've heard from earlier speakers that we've been up at, say, 40% mortality rate, but we're not seeing anything like that in this veteran population. And that whilst the mortality rate appears to be increasing, it's still a very short time period to be able to say very much about that. And 2% is actually not too huge at this point in time. Our studies that we've made about veteran populations, admittedly most of the data we have is related to oak and to beech, would expect us to have a mortality rate of between, say, 1 and 2%. So this is not really completely terrible, considering that we're actually dealing with a pathogen. So I guess this is stating the bleeding obvious, as we say, that trees have become more affected over the 4 years. Now, the reason I present that is because we wanted to check that statistically, because we wanted to be able to also see if it was possible to identify if there were any differences in the pollards, the lapsed pollards, and the maiden trees. So you can see very clearly that the trees have become more affected over the course of the 4 years that we've studied these trees. It's also interesting to look, I think, movement of the trees between the classes. I had a sore stomach when I first looked at the data because some trees had got better. And I just thought, surely that's not possible. Maybe I've done something wrong in my field work. But actually, I was most reassured to hear other researchers say that, in fact, that is true. You can see an improvement, at least, in the health of the tree, or in the apparent health of the tree. So some trees have actually looked, and this year in particular, many of the trees did look a little bit better. However, 10 trees that had no symptoms in 2009 are actually now dead, and 21 trees have moved from no symptoms to being pretty heavily affected. But the most common movement was from being symptom-free to having mild symptoms. That's the kind of general picture. So some of the other results, we find a significant relationship between the degree of symptoms and girth. And we found that actually through all 3 years of the study, larger girth trees are less affected. And this year, there were in fact no tree under 140 centimetres that was healthy. I think that's just an interesting observation, and I'm throwing it back to you researchers. We need more work to understand some of these results. We also found in 2011 a geographical gradient, as in trees further to the east had more symptoms and appeared to be more affected than trees to the west. That gradient has actually now disappeared, which I think fits with the picture that it's moved from the east to the west, that in fact now we're seeing that it's spread, properly spread throughout the county in which I live, and given that we're furthest west in Sweden, I think we can be safe to say that it's well and truly across the country. Then we also, this year, collected some data on shade and grazing. It was based on a conversation that I had with Ted Green regarding grazing and the animals pushing down the leaves into the soil and therefore causing more breakdown of the leaves, potentially meaning that there's a lower spore loading or fewer fruit bodies. Combining that with if the trees were in a wooded environment where perhaps the spores wouldn't reach it quite as easily, we did a very basic thing once again. It was semi-shade, open, or shaded, grazed and ungrazed, and we couldn't find any significant differences or any pattern in that data with all the different correlations. Interesting, and this is just, I think, a classic photograph of what ash dieback looks in the countryside, that you will see trees that look virtually symptom-free next to trees that are virtually dead and some in between, even if they've all probably been exposed to the disease for at least a decade. So pollarding in ash dieback, well, what can we say about that? And that's always been something that I am very interested in, but I think, as always, we probably have more questions than we do answers with regard to the results that we've collected. Now, what we have seen, actually, is that pollards appear to be in better condition than our maiden trees. And we did all sorts of clever statistics with this and tried the correlations between year, and we looked at the pollards that are lapsed, as in old pollards that haven't been cut for some time, and the group that have had more recent cutting and are probably in a regular cycle of cutting. And there was no difference between those two groups, but there was a difference between those groups and the maiden trees. Now, I had some discussions with David Lonsdale yesterday about this as perhaps the reasons why, and I think David's suggestion was that we understand pollards have a more complex system. They are often separate functional units. Perhaps that could play a role in why we're seeing the trees. The other thing that I would say, however, is that the young group, the recent pollard group, has quite a variation within that group. There are some trees which may have been cut once or twice in the period of time that I've been visiting the trees. Others have been perhaps not cut for 10 years. So there's a wider range of variation in that group. So once again, this is a question I think that we need to look into in more detail. What's happening here and why is it happening and why are we seeing these results? So just to show you that there is a significant difference between the groups of pollards and the non-pollards, but no difference, significant difference at least, between the two groups. So I think, as with all these things, you end up with more questions than answers. But I am retaining my optimism. I do wonder about old trees helping us with the future of ash. By the very fact they've survived several hundred years, they're in an environment which is presumably more optimal for them. Perhaps they have an advantage, either a genetic advantage, an environmental advantage, the soil conditions, the endophytes within the trees. I don't know the answers. This is something that I think just requires much more research. Why are they showing less symptoms? Why are the pollards showing less symptoms? But actually, as Marco mentioned, the biodiversity impact, I think, is something we need definitely to do more work on. What lessons can we learn from Dutch elm disease? And what can we do? We can't, I just feel, sitting idly by, watching our biodiversity disappear at the rate in which it potentially may well disappear is something we need to work very hard on. Mortality rates, the comparisons and expectations, I think that's also very interesting, that our old trees are not dying as quickly as the younger trees. And is that just a factor of time? Or are there more complex issues? The geographical gradient, well, obviously the disease has come from the east, but the environmental factors that have been touched on this morning before lunch, which was very interesting to me, about the optimal environment for ash, those ones appear to actually be in better conditions than the ones that are in wetter environments, for example. The data pollarding variable, I've already mentioned that. The other thing, obviously, is we don't really know the condition of the tree before pollarding in terms of how much was removed and how much was left on. Perhaps pollarding may temporarily remove the symptoms. That was definitely something we talked about, first of all, when ash dieback first was identified in Sweden, on Gotland in particular, where there are 200,000 ash and elm pollards, and everyone just felt absolutely depressed at the prospect of losing all of those trees. And this complex functional units in pollards. So I think it's fair to say there's definitely more work to be done and more research here that's required, and perhaps they do still hold some of the secrets for us in terms of the future of ash. Now, just to come back to sort of practical implications, because that was one of the main reasons for doing this work, was we had landowners that we were talking to. What should we be doing with our ash trees? Shall we chop them down? Everybody has, in Sweden, at least Dutch elm disease much fresher in their minds than perhaps here, because Dutch elm disease still hasn't actually been quite as devastating as it was here. So the idea that you would remove the trees appears to be a sensible solution. So this was a very clear focus for us, was to come up with a very simple set of recommendations for landowners. And our aims with that were to avoid unnecessary loss, at least, of biodiversity. Ensure equivalent replacement trees. So thinking about what can ash be replaced with. We've lost elm or are losing elm, but also aiming to reassure landowners, because I think that's something that I've found really interesting. Over the course of visiting the same sites, in 2009, when I talked to the landowners, they were saying, well, it's not long, they're all going to die, aren't they? And that's that. And this year, it's not quite as bad as we thought. So I think it's very interesting, this perception, how people view the ash and its value. So some of the recommendations that we have for veteran ash trees in Sweden, and perhaps the relevance for the UK, is that we have actually stopped any restoration work on old pollards, any kind of pruning work. And part of the reason for that is that we've seen, when pruning work has taken place, that the new shoots that are produced as a consequence, if the tree is already susceptible to ash dieback, they will be hit, and potentially you're shortening the pathway into the main tree and perhaps shortening the life of that tree. And you've invested money. And I'm coming at this from nature conservation, where we have usually extremely limited budgets. And so the priority for us is that we're investing our money wisely. However, in terms of the pollards, I think we've said that continue with cutting those trees if they're in a regular cycle and the trees are in good condition. Now, it may be that we suggest carrying on even with those trees that are beginning to show symptoms, because I think that will be very interesting to look at in the future. But avoiding cutting all trees in the same year. Spread the work so that we can begin to collect information on this. Don't fell, obviously. Others have said the same. And replacing with native deciduous trees. And diversity, I think, has been a word that's been used several times over the earlier part of today. And I think monitor your veteran population here too. You've got the database. An ancient tree hunt database exists. That means that it's very possible to set up something similar and invest in some research for our veteran trees so that perhaps they can unlock some of the secrets. Thank you very much.
Rimvys told me that I've been too strict with the first speaker, so we will take time for 2 questions. One one for you and one for Marco.
Okay.
But the Burmese, I've been told that you had a question for Marco, right?
I did.
Sorry.
It was if you knew if there had been any biodiversity studies in relation to the experience with Dutch elm disease.
I'm sure there are studies, but elm is also quite a special tree,
Yeah.
and so we have less knowledge about elm than about beech, for
Mm.
example.
Yeah.
Could you say there were quite a lot of studies done in the 70s and 80s on things like butterfly, white-letter hair streak, and then particularly in Scotland there was concern about the lichens. I remember going around some sites where big wych elm were being threatened, but there was ash alongside.
Exactly.
And I think people were a bit less worried.
Yeah. And now we've lost or losing both.
One question for Vikki.
Just a quick comment. If you're pollarding trees, you need to disinfect your instruments. You'll be transferring infections and all sorts of things.
Usually in Sweden, when they're doing this kind of work, it's the same landowner doing his own trees with hand tools, but it's an interesting point. So,
Okay. So next speaker will be Professor Halvor Solheim. Norway.
Yes. Thank you. I am honored to be here, but first of all I have to... Yeah, there is it. I'm a bit nervous because of these old pictures here. So, but I will talk about ash dieback in Norway from 2008 to 2013 and a bit about monitoring of the disease and also spore spread. First of all, then, we have ash in Norway, but it's not that much. 3.2 million cubic meter ash, and it's accounts for about 0.2% of the volume of the tree volume in Norway. And we have then the northernmost stands in Europe, I think, that it's up in this Trondheim area, but also further far north of the Arctic Circle, we have also spread a bit from gardens and parks. Yeah, it was we saw it first in 2008, and then it was a forest, a nursery owner who was worried about ash plants in his nursery. And he said that he had also a bit of that the year before, in 2007, but then he thought it was a winter damage. And we had really a huge winter damage from the Swedish border to this area. So it was not easy because the symptom may be very similar. So we were doing some isolations also in 2007, but we could not find it. But then in 2008, then it was down here. It was a nursery owner. We had imported plants somewhere from Sweden, and in Sweden they had the disease many years earlier. And then we were doing a survey in this area and found it in nearly all parts here where it's red here now. And we also, at that time, people was not aware of this disease, and it was a lot of plants for planting up here, taken from this area, up here in the Trondheim area. And when we inspected those, many of them were diseased by this ash dieback fungus. But those were burned, and we have not seen anything up there after that. But the year after then, in 2009, it was 30 kilometers, about 30 kilometers further spread here, and it was up in valleys here also. And the next year, in 2010, it was a further spread here, in Rogaland County. It's approximately 30 kilometers. But in the late autumn then, we had some branches where they thought it was ash dieback, and it was really ash dieback also there. I was on my way to Japan at that time for a 4-month stay as a visiting professor in Kyoto University, so I was not able to do anything, but the year after, in 2011 then, when I started to survey further north here, then I met another front who's coming southwards. Suddenly I saw also 2 years old, up to 2 years old necrosis who had been caused by the disease. So it must have been an introduction in this area. And also when I went further north, and it was less and less, but suddenly here it was also a few older necrosis, so it could have been also an introduction here near the Bergen. So then I went further north, and this year it was here, the front was in this area, just north of the longest fjord in Norway, Sognefjorden. So then I had to start again. No, I'm always in the wrong way. And in 2012 then, it was spread in this area, and further north here, and it was, approximately along the coast, it was more like 25 kilometers or 27, and a bit more than 50 kilometers here in the interior of the fjords. But we found also in a garden here up in Ålesund a tree who was diseased, but this was removed and burned. And the next year we couldn't find anything up there, but it was continuing spread in this area. Yes, between 38 and 50 kilometers further north. And now you can see that from this this map of our ash. So now it's here it's more continuous ash stands, but here it's more and more spread. So hopefully it will go slower further north. Yeah, so this was in a way the spread of the ash disease in Norway since 2008 when we saw it first time. But at least it had to be there already in 2006. Yeah, and we have some monitoring plots. Here we have the municipalities with the ash dieback, and here we have some monitoring plots. Up here we could not see anything this year, but nearby, 400 or 500 meters away from this plot, we found at least one single diseased tree. And here it's not so much either, but in this area it's now starting to be more and more. It's more and more dieback there. But here we have followed this for a longer time. This we have followed since 2009. So we have some, in the monitoring plots then we have 10 big trees, large trees, and 40 small trees around each of these big trees. And here the results then from these 4 plots. And we can see that, and this is from small trees. We can see that more and more of the small trees are dying or dead already. So while for the larger tree, it's only a few trees who is dead now, and it's going much more slower with the big trees. And of course, that's probably the nature. If you have one or a few infection spots on a small tree, that may kill the tree, but you may hardly see it in a really old tree. Yeah, we have been doing some spore sampling using a Burkhardt pollen sampler, and we have and we can find that then the spore in the air then rather soon after the fruit bodies has appeared. Yes, in late June or early July. So then it's more and more spores to July and in early August, and then it slows down again. And that's for 3 different years. And we have also used then, it's not so easy then to when you are using a microscope, because the spores are very similar with albedus spores, in Swedish albedus, but they are a bit bigger. In mean, they are bigger, at least. But when you're using PCR, then you can be sure that you have the right species. And here we have also we've been looking for albedus. It's very, very little Himmelskufus albedus in the stand where we have been tracking spores. And when we are doing this, the Burkhardt are there staying for 7 days, sampling spores all night, all day. And here is the typical example. You can see the slides, a very dark line here, around 7 o'clock in the morning. And there we can see a lot of ascospores, 5 o'clock, less, and in the midnight, very little ascospores. So if you would like to see ascospores, you have to be early up in the morning. So here you can see then it's it's a top in early in the morning, at least, and then it slows down more and more, and in the midnight it's very little, and then it's starting to be much more again. Yeah. So here we have then the ash dieback group in Norway. They are doing a lot more than I have been talking about now. We have 3 genetists, molecular biologists, and forest pathologists in the group. Yes, and also then, if you are interested in philosophy and the ecological crisis, it's you can see here a picture of this ash dieback on the front page of this French translation from German, I think. Thank you for your attention.
Thank you very much. We've got time for at least 2 questions, I think.
Did you check for root pollen lesions in your monitoring plots?
Yes, not yet, but we can find it on other sites. Yeah, but it's not that common in Norway yet.
Did you look at spore load at different heights in the tree? Have you looked at whether they're more higher or lower?
No, we have it. It was the spore sampler was placing on the ground, so it was approximately half a meter above the ground. And it was in the stand where we have been doing most of this is a wet stand, so we can see it's thousands and thousands of fruit bodies there. When you are walking, you are going on many of those fruit bodies. There are a lot of them. But of course, it's maybe a variation depending on the year also. When you have a very dry summer, it's less than when you have a wet summer, it seems.
One last question.
I think the figure of 30 kilometres a year, however, has been attributed to you, if I can put it in those terms. Is that a helpful figure, do you think? Or do some years the spread may be much greater and others less? What do you feel about that?
Yes.
Airborne spread now.
But in a way, I feel that that could be it could be a spread around 30 kilometers per year. But of course, I do not know exactly what this is. But is this caused by the spore spread, for instance, or are there other things? You can see along the roads there are cuttings all the time, and it's a lot of popping up a lot of ashes, small ashes there also, which is cutted, and those are going for many kilometers, so maybe it could be spread by this way also, but that's more a speculation, of course. But it's not so easy. Maybe we can hear a bit later more about the distance, how the spore can be spread. But it's not so easy because we have small ash trees or spread all over. So I think you have to hire a boat to be on the Oslo fjord or on some to see how far away you can catch spores.
Thank you very much. Next speaker is Dr. Anne Chandelier from Belgium.
Okay, good afternoon, everybody. So I will talk about the results of a study that we carried out during the last 2 years on the monitoring of airborne inoculum of dieback disease. Doesn't work. So, as you know now, the airborne inoculum of Hymenocipus pseudoalbidus, which is the name of the sexual stage of Chalara fraxinea, is constituted of ascospores, which are produced on petioles from the previous year in the leaf litter. These ascospores are dispersed by wind during summer, so they display a seasonal pattern of dispersal, as already mentioned. And they also display a diurnal pattern of dispersal with a maximum production in the morning. And there is something which is also very important. They display a limited viability because they are drought sensitive and non-pigmented, meaning that they are sensitive to UV lights. So our objectives in our study was, first of all, to develop a non-sophisticated spore trapping system for the quantification of airborne inoculum in forest. Second, we would like also to study the seasonal pattern of dispersal in Belgium, which has a specific climate and different from Norway. And we want also to analyze the spatial pattern of dispersal and more specifically the influence of the height from the ground on the level of ascospore and the distance traveled by ascospore from an infected stand. So let's talk about our spore trapping system, which is an in-house trapping system. It is composed of pieces of match, you can see there, which are covered with double-sided tape and connect to an electric motor rotating at high speed. And the device is supplied by a battery with a sampling rate of about 70 liters per minute and a collection efficiency of 15%, meaning that about 15% of spore which are present in the air can be collected. This spore trapping system displays several advantages compared to passive spore trapped or a more complicated, sophisticated device. First of all, it is compact and lightweight, meaning that it is very easy to use in forest. Its efficiency is not affected by change in wind speed, and last but not least, it is not too expensive, meaning that you can deploy several spore traps in your stand to increase the reliability of the method. Now, regarding the detection, as my colleague told you, there is a risk of confusion between spore of Imenocyphus pseudoalbidus and spore from another species, which is a native species. So we decided to use a molecular method, and you can see on this slide the different steps of the procedure. So the rods where the spore are trapped are collected in forest. They are submitted to a DNA extraction and to a real-time PCR targeting a specific region of the genome of the pathogen. And afterwards, the result of the real-time PCR, what we call the CT value or the crossing point, are transformed into concentration of ascospore. And then this concentration can be transformed into volumetric data based on the characteristic of the spore trap. Now, some words about the test performance of our system. It is highly sensitive. We can detect a minimum of 20 spores per spore trap, which is not a lot. It is also highly specific. We do not see any cross-reaction with Imenocyphus albidus, which is a native species and a saprotrophic species. And it is also highly reproducible. You can see on this graph the comparison between the amount of ascospore collected on two different spore traps, and you can see that the coefficient of correlation is very high. Now let's talk about the seasonal pattern of dispersal in Belgium. We install spore trap for 24 hours every 15 days from April to October at 1.5 meter high in the ground. In 2012, we selected 3 locations represented here, covering the main ash production area in Belgium. There were 2 plantations and 1 mixed forest. In 2013, we selected 2 other locations here, which display a low disease incidence because we wanted to know whether our system was efficient in such conditions. And there were also 1 plantation and 1 mixed forest. Here are the results for 2012. So you have the concentration of ascospore against the different day of collection from the beginning of April to the end of October. And you see that the period of ascospore released cover 4 months for 2 of the 3 stands. For 1 stand, this one, you see that this period extended from the beginning of May to the end of October, which is a very large period of dispersal. In 2013, you can see the result for 2 other forest stands. You see that the period was restricted to 3 months because the production of ascospore started 1 month later in June rather than in May. So we were interested by trying to understand why there was a delay in the production of ascospore between 2012 and 2013. Look, I remind you that in 2012 it started at the end of May, and in 2013 at the end of June. So we looked at the daily temperature during May and June of both years, and you can see on this graph the mean daily temperature for these stands, which were the stands under study in 2012, this curve, and for the stands under study in 2013. And what you see, it is a big difference in temperature in May, and more specifically during the 2 last weeks in May, suggesting that this low temperature in May could induce a delay in the production of ascospore in 2013. As I told you at the beginning of my talk, we were also interested by studying the vertical profile of ascospore released. And to this end, we put spore trapped on the same pole at different height from 0.5 to 3 meters high, considering again a sampling period of 24 hours and 3 collection dates in July 2013. We consider also 2 different positions in the stand separated by 30 meters. And here are the results. As you see, you have the different height from 0 to 3 meters high and the concentration of ascospore for the 3 different date of collection, with 2 positions for each date of collection. So from a general point of view, what we can say it is that the amount of ascospore which is measured at 0.5 meters is in average 30 times more important than the one measured at 3 meters high. And another important information, if you look at the experiment carried out the 25th of July, so the red run, you see that the amount of inoculum at the ground level could be very, very different for places separated only by 30 meters. So it means that the inoculum pressure in the same stand at the same date can be very different. And what we think, it is that this high inoculum pressure at the ground level might probably explain collar lesion, which is often found in highly infected stand with entry into the bark via lenticels. And in Belgium, we have more and more trees displaying such kind of symptoms. The last point I would like to discuss is the distance traveled by ascospore from an infected stand. And our first challenge in this experiment was to find a place, an infected ash stand, completely separate from other source of ash. And we found, hopefully, this place. And afterwards, we decided to install spore traps at increasing distance from this stand in the wind direction, and repeated this experiment twice. The first test was carried out in July 2012, and you can see on this graph the amount of ascospore from the border, and at increasing distance from the border, up to 100 meters from the border. You can see that we have a decrease in the concentration of ascospore, but what is quite interesting, it is to see that most of the ascospores stay within 50 meters from the stand. We repeated the experiment in 2013, and again you see the concentration of ascospore according to the distance from the stand. In this case, we put spore trapped up to 500 meters from the stand, and you see that again, most of the spores stay within 50 meters from the stand. But in this case, it is interesting to consider that even at 500 meters from the stand, we still have a great amount of spore in the air, probably because the concentration at the border was more important than in 2012. So what are the conclusions? We have developed a method combining a spore trapping system and a real-time PCR for the detection of airborne inoculum of Hymenosipus pseudoalbidus. This system seems specific, sensitive, and convenient for the trapping of ascospore of this pathogen, but it could also be used for other pathogens, and we think that this system could be used as an early warning system for other disease. Regarding the seasonal pattern of dispersal in Belgium, we found a very big difference according to the year, from 2.5 months in 2013 to 4 months, which is very a lot, in 2012, with a maximum in summer. And regarding the spatial pattern of dispersal, we found a significant trend for decreased spore densities with the 8, a high variability of inoculum pressure within an infected stand, and finally, most of the ascospores coming from an infected stand stay in the close vicinity of this stand within 50 meters. But in some situations, the amount of ascospores that escape this deposition area can be important. Okay, thank you very much. Before ending my talk, I would like to thank Dr. Milan Barak, who spent 1 month in my lab during this summer in the framework of the FRAXBACK Action, and also 2 colleagues who participate to the development of spore traps. Thank you.
So if most of the spores are remaining within 50 meters of the wood, how does that work with long-distance transmission in your mind?
Oh, sorry, I didn't understand.
Well, if most of the spores are staying within a short distance of the wood, how do you then think long-distance dispersal might be happening?
Yes, one possibility for me could be the petioles, which are a source of spread, because the infected petioles can stay, can produce apothecia after 2 or 3 years if the conditions are not suitable for the production. So it could be an option, transport by animals, by soil or something like that. You have also plant for planting, which constitute a big issue in this system. And finally, you don't have to forget that there are a lot of ash along the roads in forests and so on. So it's a stepwise spread system. So for me, it's a possibility.
One question.
Sorry? I just wanted to comment on the long distance transmission of spores. I think it's important that you consider the roots again.
Yeah.
Trunks. And there are lots of long distance transmission vehicles, so to say. So whether a spore can fly 500 meters or 20 kilometers per day or per year doesn't really matter for the transmission of the disease now anymore.
Is that the spores are drought sensitive?
Yeah.
Like usually they get pigmented as soon as they are discharged.
They are pigment when they germinate. Yeah.
Do you know about
it's different.
the time scale?
No, unfortunately, I don't know.
Maybe one last question.
Could I just say, I think you must remember air masses moving from the continent can carry spores huge distances, hundreds of miles in a few hours, can't they? So that can allow for this long distance transport. We only have to look at some of the records of many famous tree diseases, particularly rust.
But rust are very resistant. We are talking about spores which are pigmented and very resistant. It's not the case of the spore of Imenocephus pseudoalbidus.
I do understand that, but it's not only rust. Basidia spores of Heterobasidia nanosum, for example, have been detected alive hundreds of miles from the coast.
Yeah, okay.
Thank you. So the next speaker is Dr. Barbara Schulz from Germany.
First, let me see. Yeah. First, I'd like to thank the organizers for including our results on the potential use of endophytes for biocontrol of ash dieback. Isn't it supposed to go forward with this? But I'll do it with this. Endophytic fungi, if you think of the word, endo means in, phytic would mean plant. It would actually include all fungi, endophytic fungi, growing within plants. But the term has come to be used only for the fungi that grow in plants without causing visible disease symptoms. Colonization can be intercellular, between the cells, or intracellular. It can be localized, systemic, or extensive. Here, an example of intercellular growth. This is intracellular growth limited to one cell, and here, intracellular growth. This is spread within the whole roots of this little large trees. The endophytic fungi are phylogenetically very diverse. It's an ecological group and not a taxonomic or phylogenetic group, and they have multiple life history strategies. Can be cryptic infections that you never realize at all, maybe latent pathogens, or fungi that are waiting to be saprotose after death of the host. We have had industrial cooperations with industrial partners, and had found more than 20 years ago that endophytes are a very good source of biologically active metabolites. And here, a comparison showing these aren't actually endophytes, but they're fungi isolated from algae. Here, those isolated from plants, and here, soil isolates. And in the case of the ones isolated from algae and from plants, more than 80% produce some sort of biologically active metabolites in our tests. And the culture extracts of the endophytic fungi, 60% were antifungal, and more than 30% were antibacterial. So the industry says, yes, this is very good. We'll pay for the research because they're good sources of novel metabolites for pharmaceutical and agrochemical products. That's good because then you have money for your research, and that was very good for us because it gave our labs money. But the question remains, which I think is much more interesting, what is the function of these metabolites in nature? And hypothesis is with such a high proportion of antifungal and antibacterial metabolites that they may be active against competitors in the host. And I remember I read a paper in 1980, and this thought wasn't popular those days, by Alfred Demans saying that organisms, microorganisms, wouldn't produce secondary metabolites if they didn't have a function in nature because it cost them a lot of energy. So we had the idea that the endophytic fungi of the ash might produce metabolites that are toxic to Haemosyphilis pseudoalbidus. And there is a case in which an endophyte is being used because it produces, it's a Phylocephala rugoleucine, and it's being infected into spruce trees, and it produced the metabolite concentrations in planta that are toxic to the budworm. And so this was the idea behind this, that maybe we'll find one that also produces metabolites at a concentration in planta that will inhibit growth of the pathogen. So we isolated endophytic fungi from twigs, leaves, and rachis, which also the same as petioles of healthy and diseased ash trees. We co-cultured them with the pathogen, and this is what such a co-culture looks like. Here's the co-culture. Here first the monoculture of the Haemosyphilis, the pathogen. All three cultures inoculated at the same time. The monoculture of one of the endophytes. And you can see that in the co-culture, compare these two, there was quite an inhibition of growth. And this was not just the case with this one isolate, but it was the case with many of them. Here we have another endophyte, a different isolate of Haemosyphilis, and again you see here inoculated at the same time, the zone of inhibition. This pathogen has been inhibited in growth, and as in the case in the previous slide, you can see here that the metabolites must have been secreted before into the medium before there was contact between the two cultures, colonies here. So these are the sort of fungi that we're interested in, and we were quite surprised but pleased to see that most of the endophytes, and this is just an example of a few of them, significantly inhibited growth of the pathogen. And we didn't just look at one strain, but we looked at a number of strains of pseudoalbidus. And we were also interested to see, this is an ecological question, did the presence of the endophyte or did the presence of the pathogen induce the partner in the co-culture to produce different metabolites? And the metabolism, secondary metabolites that we found were indeed different. Red, so here we have monocultures, this is the medium, monocultures of the pathogen, 3 isolates, monocultures of 3 endophytes, and here the co-cultures with the 3 pathogens of the 3 endophytes in the co-cultures. And you see that novel metabolites are being produced, and some metabolites are no longer being produced. And in some cases, the concentrations of the metabolites were also varied. And one such example is the concentration of viridiol which is a phytotoxic metabolite produced by Humanoscyphus pseudoalbidus. It's also produced by albidus, which is interesting and which we hadn't expected. There seems not to be a correlation between virulence and the concentration, but this was measured in cultures. Still a question what happens in planta. But what was very interesting for us is that the concentration of this metabolite was greatly decreased in co-culture, and not decreased. I ought to explain something here. Here, each of these is for one isolate of the pathogen. Monoculture of the pathogen here is the first one, and then 2 endophytes in co-culture. And here the concentration of viridiol, and it's much, much higher here in the monoculture than it is in either of the co-cultures. ND doesn't mean not done, but it means not detected. And it's decreased in all of these pinkish dual cultures. So in planta, endophytes may not only produce metabolites that are toxic to the pathogen, but they may also influence the synthesis of the phytotoxins by Humanoscyphus. So the next step is, because we're interested in biocontrol of the pathogen, is to see what happens when we put the endophytes into little ash seedlings. And these are baby food jars, and these seeds were surface sterilized. This work was, I ought to have added Carina Juncker in her dissertation, did most of the work, also with some students that we supervised. And the chemical work was done by the group of Jeroen Dikschat and Rainer Abel. And this is an actinically cultured ash seedling. This is one that's been inoculated with the endophyte, still healthy. This is a Phomopsis. And here, this is supposed to see what will happen if we add the pathogen. Is it protecting it? And you can see it's not. Unfortunately for this research, Carina finished her dissertation. She now has a job and is not in the lab anymore, and I'm happy for her that that's the case. But we have many more endophytes, and maybe one of them, or more than one of them, will be successful. In addition to that, it's possible that the axenic culturing was just too much stress for the seedlings, because growing in our plant growth chambers with artificial light on an agar medium, that's just nothing like being in nature. So, in summary, co-culture of endophyte and pathogen. Most endophytes secrete metabolites that inhibit growth of Humanoscyphus pseudoalbidus, and the concentration of the phytotoxins secreted by the pathogen may be lowered. In the outlook, other endophytes should be tested in planta, also under non-sterile conditions. Can endophytic fungi be used for biocontrol of ash dieback? My answer is hopefully. And maybe that will be answered in the future. And I should mention the names of all those who did work here. Siegfried Dreger identified all the fungi. Christian Citroën did the chemical work under the guidance of Jeroen Dikschat, and Carina Juncker and the others listed here, except for Rainer Abel, did the experimental work. And thank you for your attention.
So we have time for some questions.
Hello. Sorry. If you are successful in finding an endophyte that is effective, do you have an idea as to how that might be able to be used in a woodland situation?
I don't think it can be used directly in a woodland situation. My personal concept would be that the seedlings, and that's what's being done in Canada with this other project, the seedlings are infected, and then of course you'd have to monitor to make sure that the endophyte remains within the tree. This is the case with this Phylocephala. And then they grow into saplings and are transplanted and planted.
Okay, so it's protecting the next generation?
No, that's another question. It's protecting that tree, potentially. Maybe it's protecting the next generation. The next step would be to see if it's actually in the seeds, which we can't do for quite a number of years. Yeah.
Where did you get your endophytes from geographically?
Near Braunschweig.
Okay.
There, we have the elm forest.
A more fruitful area for investigation might actually be Eastern Asia. Where
Actually, Mark Stadler, who's also in Braunschweig, is starting a cooperation. He's still in Asia, and he's coming back Sunday, and he's talking with the people who did the work, Chow and his group, on this Lambertella, which is actually a Hymenoscyphus pseudoalbinus, about isolating endophytes there. A problem might then be, as a thought that I've had, getting registration in the EU to introduce an exotic species. That's why that would be very interesting if we could find that, and it might be more promising. But on the other hand, yeah, what about all the regulations? Yeah.
Good luck.
I'm too old to follow, be allowed to follow on this project till it's, I think, finalized.
Thank you, Barbara.
You're welcome.
So it is 1:34, so we've got plenty of time.
Oh, we've got time for lots
For
of
20
questions.
minutes coffee break.
No, we have time for lots of questions at 1:34.